So what are the big challenges in metagenomics today? And I don't think they're big secrets, right? So everyone's been doing a lot of sequencing, but we don't have a lot of targeted medical interventions yet. And in particular, we don't have a lot of well-defined metabolic pathways. We know maybe what some of the culprits are or the players, but not entire pathways. And what is slowing this progress? So again, not a secret, a lot of microbiome samples are not very well annotated with patient medical records. The records are either not connected to the sample or they don't have the information that's really needed. DNA extraction methods are also a challenge. They tend to introduce bias because of gram-positive, gram-negative. Sequencing technology can also introduce more bias, right, because of the well-known GC versus AT differential efficiency of sequencing with most platforms. Assembly or error correction is data inefficient. So I don't know if you know, but in a typical metagenome assembly, between 30 to 70% of the data used to make that assembly cannot actually be mapped back to it for various reasons. And actually, the more complex the sample gets, the worse that statistic is. So a lot of people are thinking about, do we have all the data types that we need to really dig into what's going on in microbiome metagenome samples? And people are exploring different avenues for adding more information. So a lot of people are looking at metabolites. Can we understand what's going on by looking at that? Spatial information is also a really big new trend. A lot of things that don't really seem to have structure turn out to have a lot of structure, and that structure can give important cues about what species within a metagenome sample are actually interacting. And then, of course, there's long read information. Can we use long read information to get more from our metagenome experiments? And since this is PacBio, I'm obviously going to be focusing on that last one. How can we use long reads to improve our understanding of metagenomes? So there's 3 different ways that you can use long-read sequencing to look at metagenomes. So to run through them briefly, one is full-length 16S. And 16S is a great way to distinguish what is in your sample, who are the players. And the advantage of 16S is that it gives you species or sometimes strain-level information. And so with PacBio, you can either use our own protocol, or we have a partner, Shoreline Biome, who is here today that you can talk to. And they have some really great kits that cover every aspect of the pipeline or the workflow that we do not. So they extract, amplify, and then at the tail end, analyze PacBio data. Another approach is to use shotgun sequencing. So with shotgun sequencing, you can use HiFi reads to reveal intact genes and whole operons without even having to do assembly, right? And this lets you move beyond just who are the players and answer what are they doing. And the key advantage of HiFi reads is that you can get this information even without doing assembly, and I'll go into that a little bit more in a moment. And then finally, closely connected to HiFi sequencing, you can also do assembly. So while the HiFi reads are accurate and long enough to yield information on their own, you can assemble them further using a canoe or the assembler of your choice. And this is really great when you need to resolve the genomes of microbes that cannot easily be cultured. And I'll have some examples of that as well. And then finally, you can use that assembly oftentimes maybe paired either with Hi-C data or epigenomic data to further pull together contigs and also associate plasmids and viruses with specific hosts within your complex community. So this has been put up a couple times. HiFi reads, it's highly accurate and long. And in the context of microbiome sequencing, you can get up to 2 million Q30 full-length 16S sequences using the Sequel version 2 chemistry. Of course, it's already been hinted at, we're coming out with a new chemistry very soon, and preliminarily, it looks like we're going to be able to increase the number of shotgun reads you can get from 10 KB libraries quite significantly here. So that's not a final number. It could get better. So speaking first to the 16S method, here's some proof of concept data that we did in-house looking at mock communities. We use 2 different mock communities. One is staggered, so the 1002 sample, and the other is an even sample. And we multiplex those very highly, either 48 or 96 plex, and got a whole lot of barcodes for every one of those reads for every one of those barcodes. And so at this level of multiplexing, the application is very price competitive. And even better than that, because we don't do more amplification on the instrument, it does a really nice job of maintaining the representation of the underlying sample, which is always a challenge with 16S sequencing. And on top of that, it's also extremely consistent. So this is a graph showing the high reproducibility of our reads, showing that over the course of only 32 could fit on the screen easily and still be decipherable. But you can see that there's very consistent reporting of what is in that mock community sample. So the next way that you can use HiFi data is doing shotgun sequencing. So here we have two different sample types. One is the mock community, which is always a great place to start with proof of concept. But then we also sequenced 3 different human fecal samples. And what you can, I think the take-home message here is that these QV and read length metrics outperform many short read assemblies, right? And so I don't think that's any big surprise to anyone. Assembly is always, or getting contiguity is always easier with long reads than it is with short reads. And then a great thing about using a mock community is that you can start to answer questions about how accurate are the results. And so what I'm showing here is that on the left bar chart, that's what the data showed, the composition showed from shotgun sequencing, and on the right is what ATCC says is in that sample. And you can see there's a very high correspondence there between what is sequenced and what is not. And so that, of course, is showing the lack of bias with PacBio sequencing technology. So we've actually posted this data set online. You can go to our website and download it and play with it yourself. You don't have to take my word for it. And drilling a little bit more down into that lack of bias, the other nice thing about this mock community is that it contains a range of microorganisms that have a pretty diverse range of GC abundances. And what you'll see in 16S, which is the purple, is that in particular, you see it in the less concentrated samples. You really start to see bias when you do PCR sequencing with 16S. But if you look at the green, which is the expected abundance, and the blue, which is the data that you get off the Sequel instrument, you'll see that those continue to be quite well matched all the way down to low abundances, and that there's not any clear correlation with GC content there. Okay, so that's great. That's the data. The data looks good. What can you do with it? So what you can do with it is find genes. Right? And so this is an example of an analysis we did, which is very simple, on the 3 fecal samples, showing that you can take this data and put it directly into whatever platform you're already using to do gene discovery and use it to predict proteins without ever having to do assembly. Right? And so typically each one of these reads has between 6 to 8 intact genes on it that are error-free. And the reason that you can do this is because the reads are both long and accurate. And that's kind of the magical combination, right? So when you have reads that are both accurate and long, you're no longer dependent on assembly in order to do this type of gene discovery analysis. And because when you have to do assembly or error correction, you need to have higher coverage, right? And for these low abundance samples, it may not be practical to sequence deeply enough, or even possible to sequence deeply enough to get the coverage you need to do error correction or assembly. And that's no longer a concern with HiFi data. Anything that you can sequence, you can get genes from. Of course, assembly is, even better, right? And if you have species that are highly abundant, assembling them makes gene discovery even more powerful, of course. And so here's an example we did, taking the same 3 samples and assembling them using Canoe, and then following up on that by using one of the more common bidding tools, which is called RBS, through the Patrick website, where they have all kinds of great metagenomics tools listed. And so the way that this particular bidding tool works is that it looks in all of the contigs for a specific gene that is unique and highly conserved among microbes, and then it takes that contig that has that gene in it, creates a bin for it, right? And then it determines from its reference library which reference genome is most like that gene. And then it tries to map all the data against the references that have been assembled there, and then continue to bin all the contigs accordingly, using a reference as a guide. And so the reason I mention that is because when you look at this data, you'll see that RBS, it scores things as either high quality or low quality bins. And you can see there's lots of bins, and only a subset of those are high quality, right? But if you look more closely, the ones that are high quality, they're not just high quality, they're knocking it out of the park, right? So it's almost, the lowest here is 90% completeness, and the fine consistency is also very, very high. This is A+ work, right? So what's going on with the other bins? So since this is a reference-based method of binning, if there's not a reference that's close to what's in your sample, the bins are not going to look high quality. So I would argue that this is kind of an undercount of how well you can do with binning using long reads. And of course, the more that we sequence metagenomes and microgenomes, microbiomes, the more references will be available, and the more that this type of approach will be fruitful. So one of the big questions we always get is, can I multiplex? Can I multiplex on the Sequel II? And the answer is absolutely. And so how do you think about how deeply you can multiplex? So the goal of your experiment is going to determine how much coverage you need, and the coverage you need is obviously going to determine how highly you can multiplex. So the first question is, what is the estimated abundance of the most rare species that you want to observe? So for example, if you want to see a species present at 1% abundance, with one SMRT Cell 8-amp, you expect about 17,000 reads from a 1% abundant species with an average genome size, right? And so question 2 is, what is your goal with respect to that species? If you want to just determine whether it's there or not, you don't need many reads at all. If you want to do functional profiling, we would recommend you get about 5-fold coverage. So functional profiling to us means you're not going to assemble, you're looking for genes. But then if you want to fully assemble the genome and create a new reference, then you need about 20-fold coverage. And that 20-fold number is on the lower side of what you might be used to hearing, because these are HiFi reads, and HiFi reads are easier to assemble. Because you don't need to do error correction, it's already done. So we have a handy table here to do a little bit of the math for you. So the idea here is, given a certain level of coverage, and those numbers, just to remind you, that's the Sequel version 2 chemistry. The version 3 chemistry out in about 2 weeks is going to be even better. And this is the number of reads per sample you would get if you multiplexed. And then you can calculate out what 1% of those reads would be, or what 0.1% of those reads would be. And we just have it nicely annotated there about how many, what applications would be good or effective at different levels of multiplexing. So here's a real-world example of doing metagenome assembly. So this work was actually done at Maryland Genomics, and with Luke Talon and Bing Ma and Elias McComb. And what they did was sequence neonate preterm gut microbiome samples. And they were interested in sequencing those patients and following them and determining, can they see what microbes are associated with a positive outcome, or which ones are associated with a negative outcome. And the interesting thing from this study was that they were able to almost fully assemble 3 distinct strains of B. breve. And that's compelling because there are actually no reference genomes for B. breve to date. And HiFi reads, really, they let you drill down into this very, very well. So not only did they get one reference, but the resolution of the HiFi reads is enough that they could tell that there were 3 distinct strains. And so now I have one more example for you of metagenome assembly. So this is the cow-rumen assembly project, and some of you may have seen this paper already. This was work done by researchers at the University of Wisconsin, Madison campus, and also at the FDA. And they wanted to, first of all, assemble the cow-rumen, obviously. Cow-rumen is a big popular topic because everybody likes to eat beef, but we also really like having an environment that we can live in. And unfortunately, it seems like those things, 2 things may not be that compatible, but maybe we can adjust the cow rumen microbiome to make them less gas producing and make them more environmentally friendly. So there are a lot of efforts underway to understand the cow rumen. They also wanted to understand what are the strengths and limitations of different data types that are available to do metagenome sequencing. And so they collected data that's both short read and long read and also Hi-C. And so the bottom line is that one of the key strengths of short read approach is that they were able to collect tremendous coverage, right? And when you have very high coverage, you're able to do a very robust job of counting type applications. So it's really easy to get a good answer about what all is present in the cow rumen and at what level of abundancy. But with the long read data, they were able to get much more contiguous assemblies, right? And so what you can see here is that even though there was much less PacBio data collected, the contiguity of the assembly was much better. And in not only was the contig N50 or in contig N100 higher, the contigs present were on average 7-fold longer than with short read assembly. And so why does that matter? That matters because when you have more contiguous data to deal with, gene finding is a lot more successful. So in this case, this is just one of the classes of genes that they were they're looking for. And this is, and so overall, open read inference from long read bins were much more likely to be complete. And then despite lower coverage, PacBio was able to recover a lot more antimicrobial resistance genes, in particular resistance genes from the tetracycline family. And this is interesting because previous studies had concluded that you could feed cows antibiotics and that they wouldn't carry that much antibiotic resistance in their gut. And it turns out that was just an artifact of using an approach that was not very well optimized for doing assembly and gene discovery. And if you instead use long reads, what you see is actually there's a tremendous amount of antibiotic resistance. So the final thing they did is to fold in the Hi-C data, and this was really interesting because with this Hi-C data, they were able to associate the viruses that they would sequence with specific hosts present in the community. And I really encourage you to read this paper for yourself and to dig into all the details because there's some really compelling subject matter there. So I want to move on, though, since I have limited amount of time, and talk to you about some really exciting updates for a companion application, microbial assembly. So first of all, we are going to be coming out October 15th with some new features that are going to enable you to really leverage the higher throughput of the SQL system. We'll be coming out with 48 validated barcoded adapters. They're not going to come in kits. There's going to be an order sheet. And we are also going to come out with optimized recommendations pairing 10 or 15 KB library sizes with the best movie times to get the optimal results. And then maybe most exciting, depending on your perspective and what instrument that you're running today, we are also coming out on October 1st with a new dedicated microbial assembly pipeline. And this is something I know that our microbial community has been asking for quite some time. So this is a standalone pipeline, and it will not be optimized for anything other than microbes. And it's been tested and run against all classes of bacteria. And some of the new features that help it perform better are chimera detection and filtering before any data goes into assembly, also plasmid sequence recovery and assembly, circular wear polishing, and then some nice goodies for the very last steps, including circular rotation around the origin of replication and output files that are annotated according to NCB guidelines for submission. So this is some preliminary data showing the results of multiplexing 48 microbes on the SQL system. This is using 15 KB library and also the new microbial assembly pipeline. And so what you see is that all of these assemble very cleanly. Keep in mind, some of these microbes actually do have more than one chromosome. The Burkholderia in particular has 3, so anything less than 3 contigs is actually the wrong answer. And another great thing is that every one of these microbes has 200-fold or higher coverage per barcode, and that really helps improve the assembly robustness and also the error correction. And then the best thing of all is that when you can multiplex at 48 plex on the Sequel II system, the per sample cost goes down to just $70, which we are really excited about, and we hope you are too. So, and now a little bit more detail on how the assembly pipeline works. So off on the right side of the screen here, this is the summarized workflow for assembly. So we still do chromosomal assembly in the same way we always did. There's a seed length cutoff that is set in order to maximize the contiguity of the assembly. But then what we do is any raw reads that do not map back to the assembly, we extract them and we pipe funnel those into a plasmid assembly stage, right? And so for this stage of the assembly, there's no seed length cutoff, and the parameters are optimized in order to capture smaller genomic sequences. And it used to be you had to dig through the reads and do that yourself. Now we do it for you. And then the next step is a circular aware mapping. So this is part of the new polishing pipeline specifically for microbes. The great thing about using a circular aware mapping software is that it preserves high coverage even at the ends of contigs that have not yet been formally circularized in the pipeline. And that really helps with having consistent quality across the entire genome. And then finally, we add some nice features that help you, get your data into the state it needs to be in order to publish. So here's a little more detail about what circular aware polishing means. At the top, you'll see the coverage that you get if you use HCAP4, which uses a standard mapping tool. And beneath, you'll see this is the result of circular aware mapping. And in red, I've highlighted the fact that with HCAP, if you have a circular chromosome, HCAP doesn't know which end to put it on, and it ends up piling it all up just on the one end, and then the other end, it suffers as a result. But with circular aware mapping, you get high coverage on both ends, and you get complete polishing. So you don't have to go through and take the assembly, put it back in, repolish again after you move the origin of replication. It's just all done for you. And here's the results of the new microbial assembly pipeline. So what we did internally is we went and we dug up data sets which we knew were either challenging or we didn't do such a great job on. And in fact, I think we reached out to some of you as well to send us your gnarliest data sets, and we ran it against the new pipeline. And invariably what we found is that the microbial assembly very consistently did a much better job of closing these genomes, including class 3 microbes, which are pretty hairy. And in addition, we were able to do a much better job recovering complete plasmids. So there's a few cases where we did not get all the plasmids, and in one of those cases, there was just no coverage. These are very short plasmids, and we realized we still have a little homework to do on the sample prep side to make sure that those plasmids actually make it onto the instrument. But if they do make it onto the instrument, we do recover them and we do assemble them. One other instance that we did a miss, there was plasmid sequence, but it was not flagged as being finished because it's actually a linear plasmid. And so it looks like we might have a little more homework to do there as well. So in summary, we have put a lot of effort into persuading you that we continue to be the gold standard in microbial sequencing and that we're very passionate about microbial sequencing and we're passionate about getting you the results that you need. And in addition, we now have a really exciting offer for metagenome sequencing with the new Sequel II system. And we hope you all will try it out soon.